All right, for anyone who wasn't able to make it to class on Thursday, I'm assuming we're going to have a lesson tomorrow, but if you're not able to make it tomorrow, I'm going to be away for the next little while, and so I wanted to just make a summary lesson for this protein presentation. And I want you to make sure you're getting out the key things that will help you be successful. So when we look at proteins, we want to, again, understand how the chemistry affects the biology. We need to know the biology. What are the main functions? And the second big idea we have in this course is that proteins are king. They do everything. And so functionally, this is just kind of a big topics that they have. We can dive deeper into them, but they are structural functions. Proteins provide the backbone for a number of our structures in our body. Big thing is that proteins can direct and control chemical reactions. And our next whole lesson is about the use of enzymes and the role that they play to make sure the chemical reactions in our body happen when we need and at the speed that we need to sustain life. So they're structural functions. They act to direct and control chemical reactions. They react to transport substances around the body and through our cell membrane, as we see. And they can also act as chemical messengers, similar to lipids, right, as hormones. Some of those hormones can be protein-based. Now, in extreme cases, we can use proteins for an energy source, and we'll look at that when we get to metabolism. And this generally happens at generally higher intensity exercise when we might need a bit more energy. We'll break down proteins to get it, right? But I'd want to focus on those first four functions as we move forward. So now that we know what proteins do functionally, we want to look at their structure. And all proteins are made up of amino acids. Amino acids have the same kind of backbone, and then we'll look at how unique chemistry will change those amino acids. But all proteins are made up of amino acids, okay? And amino acids, you can see that they have some key functional groups. On the left-hand side, this is the first time we see nitrogen come to the party. Nitrogen makes up this amino group on this side of the molecule. We have a central carbon, and then there's a carboxyl group. Okay, so there's two carbons here. Sometimes students forget this second carbon, but we have a carboxyl on one side, that's gonna be negatively charged, and an amino group on the left side, that's gonna be positively charged. One thing that's new here is this R group. Okay, we'll talk about this more in a moment, but this R group is kind of just what sets apart individual amino acids. Okay, here you see this R group again, and this R group really determines the specific characteristics of the amino acid. There are 20 different amino acids in proteins that we have, okay, for all living things, whether it's human, plant, or whatever it is. There's 20 different amino acids, and the side chain, or this R group, determines the specific characteristics. It will determine the properties of that amino acid. And there's three different characteristics. As you go through the slideshow, you see that some of these amino acids can be polar, or they're charged. Some are non-polar, so they are hydrophobic, they have no charge. And some can be acidic or basic, or what's referred to as electrically charged. So you can imagine if one amino acid is polar and one's non-polar, the chemistry between those is going to affect how they react, versus if one's acidic versus one's basic. Okay, so the amino acids are critical based on their R group. You don't need to know the names of which is polar, which is non-polar. I'm not going to show them to you, you don't have to draw them. Okay, but you need to understand there's 20 different amino acids, and 8 of those are essential from the food that we get in our diet. But all those 20 amino acids come together and they determine what a protein's made up of. So here we just have a diagram of 4 different amino acids. So we have an amino acid 1, 2, 3, and 4. And those 4 amino acids, they're going to come together. And like we've seen with lipids and carbohydrates, they're going to come together through a condensation reaction. And we are going to have these bonds that hold them together. And these bonds are called peptide bonds. So when amino acids come together, they form what's called a polypeptide chain, many peptide bonds together. So we have a peptide here holding the carbon and nitrogen together, again holding the carbon and nitrogen together, and then at the end we have these water molecules. So we have glycosidic linkages with carbohydrates, we had ester linkages with lipids, and now we have peptide bonds. Okay, and all these peptide bonds will come together, form a polypeptide. Now the structure of a protein is really complex. There's a diagram here that shows kind of the complexity of a protein. And if you were to look at the cover of our textbook on the side of the room, again, the shape is really important, okay? And so these proteins, as the amino acids come together, the order that these amino acids come together in is going to ultimately determine what the shape is, because those side chains are going to react differently. So as you go through the lesson, you're going to see that there's 4 different levels of protein formation or protein structure. We have the primary stage where the amino acids come together, and that polar, non-polar interactions is going to start to get some little kinks and turns. Initially, we just call it this polypeptide chain. As these kind of interactions begin to develop, that moves us into this secondary, where we start to see some hydrogen bonds forming between the backbone of the amino acids, and we start to see some specific shapes. There's alpha helixes and beta pleated sheets. And then in the tertiary, we start to see more three-dimensional globular formations where these polypeptides will fold in on each other, and we start to see strong interactions between these side chains, maybe polar ones that are reacting together, non-polar ones that are reacting together. We can have acidic ones, basic ones, and so these side chains start to form strong bonds that form the basis for this. Finally, we get a quaternary structure where we might have multiple polypeptides come together, and those multiple polypeptides come together to form one amino acid. So here's just an example of this kind of formation that we have here. We have this polypeptide chain. All these amino acids come together. And then it's going to create a secondary structure, so we can have these beta pleated sheets, okay, kind of like a fan folded back and forth. And then we have a helix structure here. And so we have all these different structures. Here we have just kind of localized bonding, maybe hydrogen bonds. Oh, bell went. And then here we have the tertiary structure where we see these large interactions, stronger bonding between the side chains. In the quaternary structure, we could have multiple polypeptides that kind of get pushed together here to form a fully functioning amino acid. Again, it all goes back to the order that these amino acids come in together. So as you go through the slideshow, big idea I want you to get at is that the order of these amino acids is ultimately going to determine that shape. It's going to determine kind of how these secondary, tertiary, and ultimately quaternary structures form based on the order of those amino acids coming together. And that's the critical part. The chemistry of those amino acids is ultimately going to determine its structure. That structure is going to lead to its function. Chemistry drives biology. To fully make the case that chemistry drives the biology, we can see that one of the things with proteins is if a protein is introduced to heat, pH, radiation, harsh chemicals, it's said to be denatured. And denatured means it loses its ability, it loses its original purpose. That is, it's denatured. Right? If I was to cut your arm off, you would function differently. It's just going to change the way it is. And it doesn't matter if I change the temperature back. Once it's denatured, that protein is damaged. It has a unique structure, and if we change that structure, the protein no longer functions. So heat, we see this all the time with cooking. We change the structure of our proteins with pH, adding like lemon juice, different acidic things in our food. It changes the proteins. Radiation, harsh chemicals as well. So the chemistry here is a great example of how chemistry drives biology, because we know if the chemistry changes, if those bonds break, the biology doesn't work. The protein loses its ability to function. It has then been denatured. So a great example of how chemistry drives biology, the impact of those amino acids and the bonding that holds it together. Hopefully this helps, guys.